Good morning. Good morning. My name is Agnieszka Carpenter. I'm the Executive Director of BioME. BioME is Bioscience Association of Maine. We're a trade industry group devoted to advancing and growing bioscience in the state of Maine and beyond. And welcome to March 2023 BioME Virtual Coffee Hour. Today's event is themed, and the theme is medical devices. So when we think about life sciences, we obviously think of biotechnology, but medical devices is very much part of the industry for us as well. And we wanted to highlight some of the companies and developments within that subsector. We have 4 great speakers with us today, and each of these speakers will have 10 minutes. And then towards the end, we will have 10 to 15 minutes left over for Q&A. So I encourage everybody to leave questions in the chat. The chat is on. You can also use it just to say hi or to engage or connect. And before we start, I also wanted to thank all of our sponsors. All of these fabulous organizations are our annual sponsors for 2023. Thank you so much. Without you, we wouldn't be able to put together events like this one. So we do coffee hours pretty much every other month, and we also do a lot of in-person events. The next one will be on May the 3rd, and I will talk about it at the end. Okay, without further ado, I will stop my share here. Welcome everybody again, and I wanted to introduce our first speaker. Our first speaker is Dr. Jeffrey Halter, a pediatric surgeon at Barbara Bush Children's Hospital at Maine Medical Center. And Dr. Halter, whenever you're ready.
Yeah, thank you very much for including me in this webinar. Excited to see the other presentations as well. So as you mentioned, I'm a practicing pediatric surgeon. I've been in practice here in Maine for about 15 years. And among the procedures that we do are placement of feeding tubes called gastrostomy tubes or G tubes. So there's about 20 million patients worldwide that have these G tubes placed for a variety of reasons, but typically it's for patients that can't take adequate nutrition by mouth, or they need to use it for medications that they're not able to tolerate by mouth. So there's about 20 million patients with these G tubes worldwide, about 660,000 in the United States alone. Fortunately, many of those patients will go on to clinically improve, and they no longer require those tubes that had been surgically placed. And so our current standard of care in medicine is to remove that. Trying to get the slideshow to present. There we go. So the current standard of care for patients that no longer require their G tubes is to simply deflate the balloon that's holding it in place. And on this little schematic, you can see a tube that's going directly through the abdominal wall, abdominal muscles, and into the lumen or inside the body of the stomach. So they have a balloon that holds them in place. So I kind of liken these surgical sites, once they're healed, very much like a piercing, where you can remove the pierced earring, for example, and even weeks, months, or sometimes years later, that site will still have a tract that you can still access. So for many patients, they do develop what's called a fistula, which is an abnormal persistence of that site after you've deflated the balloon and removed the button and just cover it with gauze or with a Band-Aid and allow it to heal on its own. The majority of them will heal on their own without issue, but we usually give them between 3 and 6 weeks to see that it's completely closed, watertight, so there's no longer leakage of stomach contents. But unfortunately, the patients that do have that piercing that stays open, they can have leakage of stomach contents, leakage of stomach acid, and for weeks, they're waiting for it to close spontaneously. And that's currently the standard of care for taking care of these sites. So this is a couple of images of what a gastrostomy button looks like here. It's got the balloon on the inside. This is the button style, so it's got a little opening that you can plug tubing into that you can use for running feeds or nutrition into. This is a little bit different tube style, but they're all the same basic function. It goes directly through the abdominal wall. This is a picture of the balloon inflated inside of the lumen of the stomach. So that's that's what they look like typically. And then to remove them, I do have a short video that I have hijacked from the web that does show what a removal typically looks like. And then you'll see someone prepare it, and then this is a new button going back in. So my analogy of very much like a piercing, I think, is what that's intended to illustrate. And so for the patients that we remove that button and they no longer require it, we'll simply cover the site with the Band-Aid. And this is kind of the gold standard. It does have a little bit of an umbilicated or kind of a depressed appearance to it, but this is what the typical site looks like after it's had an opportunity to heal. During the healing process, sometimes there will be some leakage, and so there will be some irritation at the site. This is all pretty standard while they're waiting for their site to close spontaneously. So whenever you're doing these talks about medical devices, there's always like a sweet spot between like too graphic and not getting the point across for the pathology that you're trying to present. So I apologize in advance. Sometimes it doesn't heal the way that I had shown previously, but it heals more like this. So that was a G-tube site that after weeks of leakage and covering it with a dressing, this patient ultimately required a surgical closure. So I apologize for the graphic nature of that, but this is among the more extreme examples, but it is what we do see from time to time. And so with the interest of creating a new standard of care for how we manage these G-tube sites, knowing that there's 20 million patients worldwide, and there's about 3.5 million patients that are going to have surgical procedures to close their sites after they've had leakage. There seemed like there must be a better way for doing this. The average surgical closure after weeks of patient suffering is between $8,000 and $12,000, depending on how it's performed and where it's performed. So this was the idea behind the G-tube closure device that I have collaborated with the innovation cohort and Maine Health Innovation to bring through the patent process so far. And the device was intended to be used at the G-tube site at the time that we deflate the balloon and slide it out. You saw that was relatively atraumatic for the patient removing it. And then rather than just covering it with gauze or covering it with a Band-Aid, the device is intended to go right back through that same site, the way that you just saw her inserting the gastrostomy button, but instead of a new button, it's actually a clip. The clip also has a chemical compound called silver nitrate that is on the delivery shaft so that it will remove the lining that is kind of the pathology that causes that tract to stay open. It's skin lined. So the chemical cauterization will remove that lining, and then the clip deploys on the inside of the stomach. It's done with an awake patient in the office, takes about 3 minutes, no anesthesia required. So these are some examples that I did. Amateurish 3D printing was how I started prototyping. And I had many examples like this for 3D printing gone awry before I ultimately achieved what is this scaled up prototype that's about 6 times what you would use to insert into a patient, but it demonstrates the intent. The delivery sleeve goes into the patient, the clip comes out the end, and it is got an elastic deformability. This is intended to be made with nitinol. By pulling it back into the delivery sleeve, it opens the tines of that clip, and then releasing it from the delivery shaft and the sleeve, the clip ends up inside, clamped onto the inside of the stomach. This is not intended to be as robust a closure as a surgical placed clip with a patient under anesthesia. This is intended to tip the scales from 30% that don't heal to 5% that don't heal. So just enough to close that site so that we can anticipate that their own healing will complete that process. This is an example of our latest prototype. This is a chicken breast as a as a representation of the inside of the stomach. So this is the clip going through that site, the clip entering the stomach, and then being deployed. The tine's opened, the tine's seated on the inside of the stomach, and then this is after it's been released from the delivery mechanism. And I have one short video that also demonstrates that. This is still a very low fidelity prototype. We have been working in collaboration with the University of Southern Maine engineering department. And then with, as I mentioned, the Maine Health Innovation cohort here at Maine Med. And they have helped shepherd me through the process of the patent application and the intellectual property pathways. And we've just recently collaborated with ZSX Medical, who are a fledgling medical device company, and drafted an SBIR, which is an NIH grant collaborating with the medical device manufacturer to create the next generation of prototype and to begin testing before we can actually start using it for patients. I'd be happy to take questions either now or at the end, whatever we think is the best use of time. Thank you very much.
Thank you so much, Dr. Halter. That was super informative and fascinating. We will leave the questions till the end, although please feel free to leave them in the chat at any point. If they're fresh on your mind, you can leave them right now. Thank you also, Dr. Halter, for including the videos. I think that was very helpful in understanding what you do. Okay, so now we're moving on to our second speaker, and our second speaker is John Konsin, co-founder and CEO of Prapela. You may have heard of Prapela before. John has spoken at our, I think, annual conference in 2019 during a snowstorm. Thank you again for that, John. And I think John has some exciting developments. So whenever you're ready, you can start.
Thanks, Agnieszka. Hey, everybody, it's good to see you and happy coffee hour. So I'm John Konsin, CEO and co-founder of Prapela. And just a little bit about me, I started my career in medical devices in 1980. And for those of you doing the quick math, yes, that's 43 years. Over 35 years of my career was spent climbing the ladder at the big medical device manufacturers. And just as a reminder, what is a medical device company? It's a company that's developing a device to either diagnose or treat a medical condition described by the FDA. And all medical devices need to go through a regulatory approval process by the FDA before they can be marketed. So this is a regulated industry that I grew up in. And for lots of crazy reasons, as I approached near the end of my career, I decided to do a startup. So I kind of kiddingly refer to myself as the Benjamin Button of medical device startups. And my first company that we're bringing to market is Prapela. And we're introducing the very first mattress pad in the world that helps babies breathe. So let's talk about the need. I mean, we've all been watching the news the last 6 months or more, and clearly there's an urgent need to improve infant breathing. Between the threat of RSV and how it impacts especially infants between basically newborns up to 6 months of age, as well as preterm birth rates, which have complications related to poor infant breathing. We've got an unprecedented situation in the United States, if not the world today, as it relates to infant breathing. In fact, over 20% of all admissions in neonatal intensive care units in our country are related to infant breathing issues. And the most common issues are pretty well understood and defined by the National Heart and Lung Blood Institute. And they have indicated that the big 8 problems are related to opioid withdrawal, apnea of prematurity, which is when preterm infants aren't breathing as they should, respiratory syncytial virus or RSV, which we've all heard of, BPD, MAS, persistent pulmonary hypertension, respiratory distress syndrome, and transient tachypnea of the newborn. So in 2019, all of these conditions together accounted for about 560,000 cases of admissions in NICU across the country. And we know already, because of the impact of RSV, that this number will be well over 750,000 for 2022. But those statistics aren't fully available yet. But it's a big number, and unfortunately, it's growing. Unbeknownst to many individuals is that the FDA is only one treatment that it is cleared, that it is approved to improve infant breathing, and that's caffeine, which is used to improve breathing in infants that are born preterm that have been diagnosed with a condition called apnea prematurity. So there's only one treatment, and it's caffeine. And oh, by the way, it's only partially effective. In fact, 76% of newborns treated for apnea prematurity that are given caffeine continue to have apnea episodes. So we don't have many solutions out there as it relates to infant breathing. And it's a big concern because poor breathing can lead to low oxygen to vital organs, otherwise known as hypoxia, as well as neurodevelopmental issues. And so what hospitals and caregivers give our infants in the hospital and NICU is supplemental oxygen to prevent hypoxia. It's either given through a cannula or a CPAP or a ventilator device. Those are the common ways of providing supplemental oxygen. So our solution is pretty simple. It's a mattress. Babies breathe better with this Prapela mattress. It doesn't matter whether they're resting or sleeping on our pad, they breathe better. And it's activated or turned on by a simple touch of the button. You can see that little button push on the controller right there. There's no adjustments, there's no settings. Basically, you put the baby down on the mattress and they start breathing better. So come on, John, how does this work? Well, what the button does is it activates a vibration, a stimulation. So let me tell you a little bit about this unique stimulation that we have that was coincidentally developed at Harvard, at Mass General, and at University of Massachusetts over about a 15-year period. Well, the brain detects signals, doesn't matter if you're an adult or a kid, from both outside and inside the body that activate respiratory muscles. This is for basically our normal rhythmic breathing that we don't think about, called autonomic breathing. Well, those signals from the lungs and chest are sent back to the brain. So it's a loop, and that creates rhythmic breathing. Unfortunately, in newborns, sometimes the immature brain can't detect weak input signals, and that can cause disruptions to their breathing or irregular breathing rates. Sometimes their irregular breathing is too fast, sometimes it's too slow. But what the science of stochastic resonance teaches us is that these weak signals that are being received by the brain can be boosted by adding a new random signal, sometimes called noise, according to the engineers and the scientists. And so what Prapela does with this unique vibration is we're introducing a unique, random, noisy signal that boosts the signals, which in turn improve breathing rhythm in newborns. Our stimulation doesn't wake up infants, it doesn't disturb their sleep cycles, and because it's random, it's non-habit forming. So it's providing really a breakthrough drug-free treatment ideal for common breathing problems in newborns. It's part of the reason why the FDA has classified our device as a de novo, meaning there's no predicate device out there in the world that does what this device does. So we aim to basically become the standard mattress used in a hospital bassinet or an incubator or a radiant warmer. And I think most heartwarming for me as the Benjamin Button of startups and medical devices is that parents keep asking after their infants are utilized or using our mattress in clinical trials, parents keep asking, how can I get this and how can I take it home? So we like to say that over the last few years, we've been the most award-winning pediatric medical startup in the country, if not the world. We have our marketing clearance pending with the FDA. In fact, I was working on questions that we received from the FDA over the weekend. So we hope to have clearance on our first device soon. We've received what the FDA calls a breakthrough device designation 2 different times, once to treat opioid exposed newborns, a second designation to help preterm newborns with apnea. We have 7 peer-reviewed studies published on our technology. It's currently in clinical trial as well at Maine Med, thanks to Dr. Jamie Fay. That study is being led by Tufts, and it's also being conducted at other sites, including the University of Massachusetts in Worcester, the Cleveland Clinic, Lowell General Hospital, and others. What we've done is we took a device that was originally developed as an investigational device at Harvard, UMass, and Mass General, and we've made it a commercial-ready device. We're just waiting for basically the FDA to say, you're good to start selling this device. And we're excited that that may happen soon. Also, we hope to share some very good news in the next week or 2 as it relates to our work in the area of treating apnea prematurity. So that's it. That's Prapela, the world's first mattress to improve infant breathing, and look forward to any questions that you might have at the end of the talk today. Thanks.
Thank you so much, John. Amazing as always. I just want to say, back in November, we went to Medica, which is the largest medical device show in the world, trade show, and we had so many people stop by our New England booth asking for the baby mattress guy. So this is big. And wishing you all the best, John. And any questions, please leave them in the chat. And now we're moving on to our next speaker. Our next speaker is Dr. Mary Hayes, co-founder of Activas Diagnostics.
Thank you. Let me just get my PowerPoint up here. Oops. Hi, everybody. My name is Marie Hayes. I am I have been CEO and shared CEO with my collaborator, Dr. Alia Betty. Activas Diagnostics has been in business for since 2010. It's built on 30 to 40 years of research, many of it in pediatric settings, many of it with infants using a mattress device with apnea of prematurity, John. So I'm familiar with your application, seen it in an NIH study section, and very impressed. Thank you for that. Ours is a little different. So we've done a lot of pediatric work. We've also now moved on with NIH funding for our SBIR phase 1, phase 2, looking at an under-the-sheet mattress system for detection of early Alzheimer's disease. And as you know, Alzheimer's disease is of epidemic proportions because of biomedical advances that have caused people to live much longer, but maybe the condition of the brain in neuroscience is so complex, we don't have as many treatments for this condition. So I'm going to talk today about early diagnosis for Alzheimer's disease, because like any disease, early diagnosis has the opportunity of making significant impact, but later in the process, less so. So if you can look at this graph, these are the changes that occur in Alzheimer's disease. Usually around age 60, the rates of the normal process of aging become noticeable, and there is a decline in what we call amnestic performance or memory performance. Mild cognitive impairment is a condition that is associated with memory loss. And amnestic MCI and memory problems are often not detected. In fact, only 57% of primary care physicians and providers inquire about memory problems, which are also compounded by sleep. And you probably realize that sleep and memory are intimately linked. We'll talk about that in a minute. But as the cognitive decline persists, it can actually increase in speed, even though the disorder lasts for some 6 to 10 years. It's a very, very severe and very difficult for people to deal with. So how do we detect it in early stages? Biomarkers are being developed, certainly amyloid beta and tau. These are actually require cerebrospinal fluid at the current time. The hope is to make this a bloodborne analysis. We also can use MRIs, but usually by the time we see these brain changes in atrophy of the brain, the person is well along in the disease. We can look at PET, positron emission tomography. We can use that to look at amyloid, for example, and tau, one of the markers. But these are very invasive tests. We can do neurocognitive testing, which is very, very time consuming and expensive. So these are the ways that we do early diagnosis. As you can imagine, these usually occur when the patient is in this blue zone. So what we're hoping is that we can get back into the red zone and see if we can see these changes in primary settings very, very early on. So in the next slide, I'm going to talk about our device. So this device, again, has a very long time of development, but I'm going to talk about the state of the device at this time. It's comfortable, it's ultra low power, it is a mattress style. There are more sensors around areas where movement is increased and respiration is increased, and we are detected, and we are trying to detect those two signals in particular. The sensitivity and accuracy is much greater than any actigraphic device, Fitbit, anything that is available in the market, both as a biomedical device or as a consumer device. We say it's very low cost, and we can demonstrate that perhaps in another setting. So over the course of the last 10 years, we've been trying to measure sleep in the home with our device, looking for, as you can see in this bottom panel, these changes in blue that are related to movement, which we've found to be coupled to respiratory changes in red. So the frequency changes of respiration actually increase, which is very, very useful for individuals during sleep, because autonomic nervous system generally pushes down respiratory variability, and this can have cardiac and other effects. We know about disorders of sleep like apnea, for example, which are associated with excessively long pausing. We are not detecting apnea at this point, but we have this in our hopes in the next few years. We have tested about 100 patients with our new device, and we are able to do it in the home. And so the Activas sleep test is really when the individual comes to the primary physician or provider and says, I'm really having trouble sleeping. That should be a red flag to the primary provider to order tests. And in this setting, we have actually tested our Fourier delivered mechanism of taking the plug and play mattress device, putting it as an overlay on the mattress, delivering it to the home so that the aging person and family members can use an overnight test. And the overnight test is can be one night, can be 3 hours. We can get this information very, very rapidly. And we've developed an artificial intelligence risk score that I'll talk about in a minute for Alzheimer's disease specifically from these 100 patients that we've tested, many of them having formal diagnosis of Alzheimer's disease from Dr. Cliff Singer's Mood and Memory Clinic at Northern Light. So let's talk a second about the AI-driven threshold for early Alzheimer's disease and what exactly technically are we talking about. So Alzheimer's disease is associated with sleep disorders. These sleep disorders are generally in continuity. We call this wake after sleep onset. Sometimes it's in sleep onset as well, which we found to be associated mostly with depression. But aging people do suffer more from depression. But the disorder that is most associated with mild cognitive impairment is this characteristic of poor sleep for a considerable period of time. And we propose impacts memory consolidation during sleep and also during the day. And that over time, the brain is affected because these conditions may create chronic hypoxemia. So what do we mean by that? Well, we have this idea of an arousal coupling index, which takes arousal type movements. These are micro movements. They are not detected by Fitbits or actigraphy. And they're very, very high frequency. They have rhythmic properties that are related to essentially a 2 to 3 minute rhythm. And we're able to identify them, and we're able to look at their association with respiration, which is simultaneously recorded in our system. So using engineering methods and also very advanced statistics, my collaborator, Dr. Ali Abedi, has applied artificial intelligence. He's developed a software system with our software architect. And we basically take characteristics of training patients on the mattress, those with mild cognitive impairment, those who are normal controls, based on a deep cognitive assessment, and actually diagnosed formally by Dr. Singer. And based on those data on respiratory and movement, we were able to identify, using neural networks and other techniques, a comparison with physician diagnostics of the characteristics of movement respiratory coupling. Again, the idea is that movements are natural process in the arousal system that is impaired with sleep deprivation. So when it's impaired, it actually leads to respiratory low variability, not that increase in variability that is so healthy. So we find one of our metrics called time latency between this coupling mechanism is capable of giving us a binary distinction or classification between MCI patients and normal controls. We've presented this to the FDA, and what we are essentially claiming is that we are unique, and the FDA has determined that we also, John, are de novo, that we are a home. SleepMove is a home device. It's exceptionally comfortable, accurate, sensitive, etc. It's very different from sleep labs in that way, and it's also much more accurate and sensitive to real sleep disorder than what's available on the consumer market. Of course, this aspect of comfort, it cannot be underestimated. These are the way the devices are recording movement of respiration currently, typically in sleep labs, sometimes at home. You could see in the first case, it's very hard to sleep with that device on your head. Our executive team is myself and Dr. Abedi. We also have an engineering team, Ms. Esfahani and Samaya Kazaro. Dr. Kazaro has done the software architect work. Our clinical team is also matched by a data management and operations group. This is just a small picture of the things that we're doing, but the FDA and regulatory leadership has been outstanding. We're looking for ways to access currently existing ICD codes. We've put a lot of work into this. Our intellectual property is in development. We have patents, et cetera. And marketing and business scale up has been planned with Ted Newell of Medical Device Success and other individuals. So our milestones 2018 to 2022, we've reached intellectual property, clinical studies, publications, and play. These publications, one is going to be published next month on the AI piece. The other clinical study we expect to be accepted in the next few weeks. Our de novo status and identification of manufacturing partners. This business model is just for a snapshot, but basically we've completed our phase 2 clinical studies. We're very deep in our marketing plan and FDA activities. We have a pivot for a consumer device in mind. The device and report use for clinicians. We have plans for additional work with primary physicians at Maine Medical Center. We hope to work with Dr. Neil Konsin. In addition, we have another application of our system that NIH is inquiring about, and we're not free to talk about it this time, but it's a related disease process. Just an acknowledgement, it takes a village. This is just some of the people that have helped us and the funding involved. Thank you very much.
Thank you so much, Dr. Hayes. And I see Dr. Abedi's with us too, so welcome. Thanks for joining us. Once again, please leave questions in the chat. And last but not least, we have our fourth speaker today, Derek McKenney, Director of Engineering at Puritan Medical Products. So the three companies we've heard from, or three devices we got the opportunity to learn about, were sort of smaller scale, but we also have a very large medical device manufacturer in Maine. So Derek will talk about their company.
Hi, good morning, everyone. Thanks for the opportunity to be here today and speak a little bit about Puritan. So we've been around for a long time here in the state of Maine, and somewhat unknown. It's a very small company, and we've played a part in diagnostic testing for a lot of large companies as an OEM manufacturer, and like I say, gone a little bit unknown in the state of Maine prior to COVID. So today I'd just like to talk a little bit about what we do, what happened during COVID, how we were able to respond, and where the future of diagnostic testing is going more generally. So just a short history. We were founded in 1919. We had our 100th birthday here a few years ago. The company was originally a toothpick manufacturer in Michigan, relocated to the state of Maine in the 20s to be closer to white birch resources. Obviously, wood toothpicks and supply chain issues were alleviated being closer to northeastern forests. Now we have really 5 market segments that we're serving, the largest of which is diagnostic specimen collection, which is essentially the swabs that we all became very intimately familiar with during COVID. But aside from that, we also have industrial divisions where we focus on controlled environments. Those products are often used in the assembly of medical devices. So it's not uncommon especially with pacemakers or with implants, that there are cleaning processes that need to take place during assembly, and that's where we support. A large portion of our business is also in the forensic field, whether that's crime scene investigation, something that's become quite popular the last few years as far as genetic testing to understand, family history. We've partnered with several companies on that front as well. During COVID-19, we became very clear that there was a shortage of test kits and swabs. So in very early 2020, the White House reached out and we began our process of scaling our manufacturing facilities. So over the next 24 months, we opened 3 facilities. This effort can't go undersold. It was a tremendous effort and very much a testament to the state of Maine and its capabilities. But in that time, we performed over 300 process validations to bring our manufacturing capabilities over 400 million swabs per month, and step up to support the nation when it needed it most. Coming out of COVID, everybody started to become very familiar with diagnostic testing at home, testing routinely to know if it was going to be okay to go interact with people, in really determining the course of action from that test forward. So we've been innovating for a long time here at Puritan, and part of what we've done is really refine specimen collection. So testing is great, but if the sample collected, if that process doesn't go well, it's not effectively released, the test can be inaccurate. It can impact, again, patients' well-being. And what we want is to make sure that we're collecting the highest quality sample possible and we're releasing that to the assay as effectively as possible. So over the last few years, Puritan has developed 2 new product lines. Both are best in industry for collection and extraction. The first being HydroFlock, which currently holds 18 patents, and second being Ultra, which we have 15 patents for at the moment, and 2 more patents pending on these product lines. So they really are the best specimen collection devices on the market. And I think that's been representative with market acceptance and migration towards flock products for collection. But because we've been, again, sort of a little bit unknown here in the state of Maine, what we do mainly is OEM manufacturing. So we are working for those large medical device companies that have come up previously here today, and we're providing their specimen collection. devices, in this case, swabs. And what that really means is the future of diagnostic testing, as it changes, we're changing with it, and we're innovating a little bit ahead of it. So COVID brought this, over-the-counter at-home test to us. Companies like Q Health, Abbott released Binex Now over-the-counter. Quidel had a similar competing product out, QuickView. And now we really start to expect to be able to perform diagnostic testing in our home and have an instant result. And that's really driving the trend towards self-collection, which has its own inherent risks and really changes how we as medical device manufacturers manage patient risk. It may seem somewhat benign that an anterior nasal sample to be taken at home, but there are far more risks involved with actually performing the test. So where we're going is really working with other large organizations to develop new product lines, new processes that are very easy to use, very also easy to integrate into the home, right? We don't want to launch tests that require a substantial amount of infrastructure beyond what the average person may have, and something that's very much a low cost, quick check for some of these very common diagnostic tests that have traditionally required a visit to a physician. And I really think over the next 3 to 5 years, at-home testing will become the standard, not only for things like COVID and flu, but strep and the like. And there are many, many organizations, Puritan included, that are striving every day to bring healthcare into the home, close to the patient, and make it really as convenient as possible to understand what the situation is with the individual person, and so that they can seek the best treatment as fast as possible. And that's really in a very short summary there, kind of where we are, where we've been, and where we're going in the state of Maine with medical devices. Thank you.
Thank you so much, Derek. I think after COVID, everybody's familiar with your company. Hopefully we won't have another big pandemic like that. But as a mother of 3 young children, I love the idea of at-home testing for every possible thing. So thanks again. Okay, so now it's time for our Q&A. I do see some questions in the chat. Also, feel free to still type them in, or you can raise your virtual hand and have an opportunity to ask a question in person. So I'm just going to go through the top here from the top. I think this is for Dr. Halter. Could you please discuss the regulatory hurdles in getting this device cleared by the FDA? Will a 510(k) be sufficient?
Yeah, so thanks for your question. I don't know that I'll be able to answer it very satisfactorily, because that's definitely not my area of expertise. I'm the one that had the idea for the device, and I'm relying on my collaborators regarding the FDA moving forward. I do know that ZSX Medical has experience with testing for their biomedical devices in the past, and so I am relying on their infrastructure for moving this device through the FDA and the regulatory pathways. So sorry, I don't have more information than that.
No, that's totally fine. And actually, there's a question here that's a little broader and to everybody. So most of our questions are about FDA. I think if you have a medical device company, that's step one for everyone. But what have you done to navigate the FDA process? What was that knowledge you have on your team, or have you engaged consultants for this? What about for product testing, for efficacy, safety, electrical? Sorry, there's a lot here, but basically navigating the FDA process to everyone. Whoever wants to start.
I'm happy to start.
Sure.
As soon as our SBIR phase 2 in 2018 was getting organized for the clinical studies, we immediately started an arm of our work to commit people and resources, sometimes as much as $30,000 or $50,000 just to kick off the process of consulting with high quality consultants. We looked at Hyman and colleagues from Washington, D.C. We worked with them for about a year and a half. We also contracted with Jacob Drabkin, who was actually specialized in CMS, which, as you know, is Medicare and Medicaid services. So that process was, of course, critical. You want to have FDA and CMS in coordination as you go forward in your FDA work. I know that some of us have done considerably more than we have. We've been in finished pre-submission. We have our questions to answer, and we had a de novo status. Our next stage now is going to be to fulfill the additional testing that the FDA has requested. And this could cost, a lot of money without giving numbers. It depends on what your device is, how much it's going to cost. But in our case, the studies are very expensive. So we've started to reach out to collaborators who may be able to use our device in a clinical setting without significant charge to us. That's kind of the stage we're at. Our next goal is to increase diversity, because that was our main critique. We argued that Maine was a diverse state in socioeconomic terms, and that was really the basis of diversity generally. And that did not sell because, as you know, there are very strong, important differences between groups. And so those can cause differences in the response to your medical device. So I'll let someone else have a chance. That's what we've been up to.
Thank you. Thank you so much. John?
Yeah, I'm going to add to Dr. Hayes' comments based on our experience and kind of what I've lived through in my life. First of all, if you are new to working with the FDA, my strong advice is develop a collaborative approach rather than an argumentative approach with the FDA. You'll go much farther with the FDA with that type of approach. And I've seen it time and time again in my career. The best companies partner with the FDA. And they don't fight the FDA. The second thing I want to say is unique to us, but not unique to many companies that are in the area of developing new innovations, is to pursue the breakthrough device designation program that's offered by the FDA. There's several hundred devices that have received FDA breakthrough device designation since the program started in the last, I want to say 7 or 8 years ago. And what a BDD does is it allows you to have informal, supportive discussions with the FDA to discuss many different matters, including your regulatory pathway, your data development plan to support your indications for use of your device. So in other words, it's a softer way to get started with the FDA than to go through, say, a more formal pre-sub-submission and then using consultants up front. So if you're not familiar with the BTD program, Breakthrough Device Designation, Google it, check it out, and see if your innovation, your device is something that could benefit by participating in the BTD program. And it is rigorous. You have to apply and you have to meet certain standards to be designated as a BTD. And at Prapela, we've been fortunate to receive 2 breakthrough device designations, and that's really helped us with the FDA.
Thanks so much, John. So Derek, I would imagine you have in-house staff for FDA?
Yeah, so and this gets a little, obviously we have some much more higher class devices here around this table than what we're making with specimen collection, single use, sterile and non-sterile devices. So the one thing I would add is that, the regulations vary drastically depending on the risk associated with the product. And in our case, a lot of what we're manufacturing in the eyes of the FDA is lower risk, and we are subject to fewer requirements around our products. Not to say that there's any less work, but being a single-use disposable does come with some lesser requirements in certain areas. But yeah, we have a full regulatory team in-house. So we've been doing this for a while. So we've had the chance to build that expertise over the years as well. We know our products well, our markets well, and have a good relationship with the FDA.
Perfect. Thank you so much. I see that Dan Gray from Gray Optics has his hand up. So Dan.
Good morning, everybody. Thank you, speakers. I really enjoyed it. Sort of a broad question just about the state of the industry and med device and new product development. With our current economic situation, there's a lot of pressures on hospitals, and it affects new product development for sure. And the themes that I'm hearing about are sort of staffing constraints in hospitals as well as cost. So just curious to get some feedback from the speakers about how cost of medical device impacts new product development and what that means for the next 18 months or so, and how is that impacting things from your perspective?
Thanks, Dan.
I'm happy to answer at least initially from mine. So my device is still a ways from being available in the market, but the intent, while it's the primary intent was to figure out a way to take better care of patients so that they're not suffering while they're waiting for their surgeries. But the reality is, if this device comes to fruition, there'll be millions of patients that will no longer require admissions to hospitals, no longer require anesthesia for their procedures. So it could be a tremendous savings in terms of workforce and resources in the hospital setting if this is something that can become an exclusively 3-minute no anesthesia office-based procedure. So that is definitely first and foremost in our thinking about bringing this product to market.
Thank you, Dr. Halter. And Dr. Hayes, did you have a comment?
Just that I think that one of the questions that Daniel brought up that I thought struck a nerve was, what is the cost of getting through the FDA? What is the cost of these clinical trials? And our estimates are frightening. We sometimes, we've already spent in clinical trials about $1 million. That's not very much. That's because we relied on everyone that was a colleague for 20 years, and we're going to now jump in again. And going back to NIH, the funnel decreases as you continue to apply to them for SBIR follow-ons. And they're very strict in the sense that I think there are almost very few that go on to the last 3 grant opportunities. So because of that, you're pushed into the market to look for funding from angels and other sources. And that's a very difficult process, which as university professors, technical people doing science, that's a whole different bailiwick. So you mentioned, Daniel, how do you do that? So you have to find the business people, you have to find the experts, people like John that know how to, and Derek that already know the hoops that are going to be necessary. Also, in particular for FDA, you need FDA specific consulting, and that's very, there's nothing, even within their FDA portfolio, they have very small components with people with unique expertise for a device like yours and make recommendations. It's a very, very long process. We've been told maybe a couple more years to get the process completed. So that we've already been at this a long time. So I don't know, but it sounds like John's approach and Derek's approach and Jeffrey's approach of working with other universities and groups is what we do too. And that helps because there are other resources at larger universities and medical schools.
Great, thank you so much. Any other comments on this? No, okay. I also had a question for Derek about at-home testing. So you mentioned that you predict that at-home testing will be more prevalent. There will be more at-home tests in the next 3 to 5 years, but you also mentioned some risks associated, and I was wondering what those risks are.
Yeah, so there are certain things that happen with a diagnostic test. For instance, dispensing a certain volume of liquid accurately in a certain location, which when that's done in a controlled setting with a trained professional, that's different than presenting that to someone, in their home. And the level of confidence, again, now with COVID, I mean, at-home testing, everybody has sort of jumped in, right? And I think we kind of went through a little bit of a learning curve on what the expectation is for some of these lateral flow tests. But, historically, things of that nature have caused additional risk because if they're not performed correctly, they impact the result of the test. So the same with sampling, the depth in which a nasal sample is taken. How do we ensure that someone's not going to injure themselves by trying to put a swab 3 inches in their nose versus, a centimeter? And that's, there have been ways around that in the past, but there's design intent that has to happen to ensure that the patients are protected. It's a little different approach, different risks, and different unknowns that have to be managed.
Okay, great. Thank you so much for explaining that. Okay, we are pretty much out of time. If I missed your question in the chat, apologies. This event has been recorded. If you're interested in obtaining a recording, it will be emailed to everybody after the event. Please feel free to share it. It will also be available on our YouTube channel. We do have a YouTube channel. All of our coffee hours are there. So I think this was a particularly interesting coffee hour, and we would appreciate your help with spreading it beyond just this Zoom room. Our next event, as I mentioned, is in May. So May the 3rd is our BioME Student Showcase, which is a competition for students, but it's somewhat open to the public. If you're interested, it's in Bangor this year. Or if you have any kids or friends who are either college or high school age and they're passionate about science, application process opens soon. We will also have our Women in Bioscience in May. Stay tuned for the date. If you're not subscribed for our emails, you can subscribe easily on our website and get all the information about our events. And I want to send a warm welcome to all of our speakers today. We're getting a lot of feedback here in the chat saying that the presentations were amazing, and I agree. And thank you to everybody who joined us. And I wanted to wish you a pleasant rest of your day. For once, the weather's good today, and hopefully till the end of March. So see you next time. Thanks again. Take care. Bye, everybody.